On behalf of BioIT World Global Symposia Series and our sponsors, Certara and Collaborative Drug Discovery, I'd like to welcome you to Best Practices for Small to Mid-Sized Companies to Advance Drug Discovery Programs. My name is Heidi Brown, and I am the host and moderator for today's event. Now, I would like to introduce our presenters for today. David Lois, Executive Director, Scientific Informatics at Certara. Our second presenter is Charlie Weatherall, Director of Customer Engagement, Collaborative Drug Discovery. Welcome, David. The webinar is yours.
Thanks, Heidi. Okay, so let's dive in. We can talk about something that doesn't involve elections for an hour, so that's always pleasant for everybody. But what we wanted to do today is just introduce you to work that Certara and CDDVault have been doing together, implementing both our concepts of what constitute best practices in terms of research and particularly data flow during the research process. In terms of the agenda, we'll start off with just some basics. We'll talk about the drug discovery process, how that is inherently data-driven, and how to improve the efficiency and effectiveness, and most importantly, the speed of the discovery process. We'll then talk a bit about best practices and informatics transitions, especially when it regards the informatics in small to medium-sized organizations, which have their own particular challenges. Charlie will talk then about CDDVault and how CDDVault is used for capturing scientific data throughout the process. Then we'll talk about G360 and how G360 is employed as the analysis portion and design portion during that process. But we'll actually spend the majority of the webinar actually seeing these things in action. So Charlie will take CDDVault and show you what CDDVault does and how data is captured. Then we'll see how that data can be analyzed in D360, and we'll then complete the cycle to show how compounds designed within D360 can be loaded up into CDDVault. So moving on from the agenda, let's take a look at the drug discovery process. So overall, a key element of the drug discovery process for small molecules, but also for peptides and oligos, is what we refer to as the design, make, test, analyze cycle. And the idea here is that during the make portion of this, we are making new chemical compounds in order that we test those and see what kind of bioprofile they have, see if they have activity at the receptor of interest. So making compounds is not enough. We also have to test those compounds. And these are both data in portions. We're doing experiments, we're running syntheses, and we're pulling data into that system. In order to design new chemical compounds with improved bioprofile, we need to analyze that data. And this is where we bring the data out. And we want to bring various analytics tools and data visualization to bear so we can make the best possible decisions knowing what kind of data we have going into this. So as you can see, we are inherently data-driven. We are making new compounds, we are testing them, we are analyzing them, and finally the last stage is then to design new compounds on the basis of what you see. Now, this is just one fundamental key workflow of the drug discovery process. And there are many other adjunct workflows, for example, looking at logistics of who's made what, especially in outsourced research. Thinking about how assays are stable over time, and many other logistical and scientific workflows. But today, we're going to focus on design, make, test, and analyze. So when we look at the overall drug discovery process, it takes many months, years potentially, to get to a compound that has the appropriate physical and biological properties that we are looking for in order to become a therapeutic agent. And over this time, this period of research comprises multiple design, make, test, analyze cycles. And during this, we're not just trying to get a compound with the right kind of activity, we're trying to get a compound with the right kind of bioprofile. And that usually involves multiple assays, multiple calculated properties to say if the compound is likely to become a good therapeutic agent of the profile that we desire. As we've already discussed, data is fundamental to this. So if we can improve our data and analytics capabilities, we can reduce the time taken for each design, make, test, analyze cycle because the data is readily available as soon as it is created. Also, we can reduce the number of cycles required by making each one consistent, and also making use of all of the data that's actually available at that time, all the data that's been measured, and not have to wait for any informatics. And the goal of this is to get to our preclinical safety testing much, much faster, which also gives us a faster time to market and an overall cost reduction in the amount of effort it takes to get there. If we want to look at this a little more graphically, we can think about that same process, but now with shortened cycles and with fewer cycles, because we are making better use of the data. So now that we have a view of what it takes in order to design new chemical compounds and the overall process in getting them to the next stage of drug discovery and development, let's look at a few of the things that both CDD and Certara have learned over the years we've been involved. So we're going to take a look now at what the best practices are, and then some specific challenges to smaller and medium-sized organizations. At Certara, the D360 team has been involved with informatics for over the last 10 years. Just when you think you've seen everything, new issues and differences come about in the way people are working, in the data that they have to operate. And we've developed a series of best practices. So the first of our best practices is you really want to eliminate as much manual data collation as possible. This allows us to focus on the science and get rid of all of the busy work of moving data perhaps between different spreadsheets into and out of different systems, utilizing different applications. So by focusing on the science, we not only shorten the process of doing the analysis and coming to conclusions and decisions in the research, but we also eliminate a lot of manual data errors. You know, biological data is never as clean as you would like. So it's very easy to, for example, take an average of 3, 5, and 10 if those are our IC50 values. But what do we do if we have 3, 5, and greater than 50? Different people will approach that in different ways. Manual data collection introduces errors like that. So having more stuff automated is very, very important. If we move on, we also want to use the data real time. Certainly with D360, we've spent much time swapping out systems that people previously had, be those just the use of Excel or right up to large pharmaceutical companies replacing their in-house built systems with the productized version of D360, which fulfills all their needs. But what we've seen in those older systems, especially Excel, or when the original systems were perhaps not as efficient as they could be, then the data could not be used real time all the time. And that means you're making decisions when you know compounds have been tested, but the data is not accessible easily enough to take advantage of it. And this, again, makes the drug discovery process much longer than it should be. We also want to ensure consistency. So by thinking about how data is captured, at the same time as you think about how it's going to be used, you can ensure that that data is captured much better, that it's used much better, and also that it's used consistently. So this again is more of an automation concept, but it's also when you're considering a change in your informatic systems, and especially when you're considering getting into the first, what I refer to as mature informatic system, and changing over from an Excel or other based system, then you really need to think about both data in and data out quite holistically. One of the things we've seen, this particularly applies to small companies, but also increasingly at large organizations. The amount of IT support varies drastically, but over the years tends to get lower and lower and lower. And so what we want to ensure is that the system, both in terms of pushing the data in with CDDVault and getting the data out with something like D360, does not require any specialist IT support. So hence the intuitive interfaces in CDD and D360 for adding and extracting data and analyzing it, but also other things like the ability that all data that's put into CDDVault is available real time in D360. When new assays come along, there is no requirement for anybody to do any work. That new assay is automatically available in D360 and therefore accessible to users immediately. If we look now at some of the organizational challenges that perhaps smaller or medium-sized research organizations have, we've seen a variety of them, but boiling them down to the most important one First and foremost, either limited or no IT resources. Now, a lot of the capabilities within software these days and their requirement for IT is helped by moving things into the cloud and having hosted systems. But that on its own is not enough. You also need the systems to be self-service for scientists. So the ability to go into an application and build your own data query or register your own compound or add your own assay results all must be done by somebody with the basic scientific knowledge and not require somebody with specific database knowledge to go in there. Secondly, one of the key elements we see, especially with smaller organizations, is when they first get into the search and they first start getting their substances and testing them, that data is often captured and analyzed and understood in Excel. Excel is probably the most commonly used research application today. But transitioning from Excel is very important because using Excel requires a lot of that manual data collation. And while that can be done initially, as that data grows in volume or as a new project arises, the workflow about using Excel will very quickly require substantial and massive effort, is potentially error-prone, and exceeds that manual data entry capacity. And so we want to have a system that is going to not only provide access to the data, but provide input of the data without all that manual effort. The third thing we see here is how people get into new systems. So when you're moving either from a rudimentary in-house system or you're moving from Excel, usually the first thing people think of is like, well, I must capture my data properly. And that's a great thought. And that's really where the CDD guys come in. They do an excellent job of capturing that data. But also when you're thinking about capturing data, it's also very important to think about how to get it out. So moving data initially into a data capture system without thinking about how you get that data out actually makes the research process harder, even though it's making a step in the right direction. So part of the reason for our collaboration here is so that the people can now very easily get both the data in and the data out all together. That improves the situation if you're moving from Excel or an in-house system. It allows you to immediately see how, when you put the data in, you're going to get it out and whether that's suitable for analysis. So this just gives us an idea of all of the best practices and also some of the concerns that small to medium-sized organizations we have. And as I've mentioned a couple of times already, this is the key reason why CDD and Certara have got together with the Vault system and the G360 system. The whole goal of this is it's all cloud deployed, it's all pre-integrated, and it's all ready to go immediately. And if we take a look back at that design, make, test, analyze cycle, we see that the CDD covers kind of the make and test portions of this. So this is all the portions where data comes in and needs to be captured, needs to be reviewed, and perhaps reported. But when we come to the detailed analysis, that's where D360 comes in on the Certara side. And here, D360 deals with all of the data out, the analytics, and team collaboration around how that data is dealt with and how new compounds are designed. And again, both of these systems have essentially zero maintenance overhead. And so they can be deployed at small organizations where you might not have IT resources, or any other size organization where even if you do have IT resources, those people are generally pretty busy, and you also don't want to wait until those until those resources are ready. You want to use the data real time because that enhances the efficiency of your overall process. So having made this introduction of drug discovery process, key principles, best practices, let me hand this over now to Charlie, and he will talk now about the introduction to CDDVault and how that works. So let me just hand over, and Charlie, I think you should have the screen.
All right. Thank you, Dave, and thank you to Certara and CHI for inviting us to participate today in this webinar. We certainly appreciate it. So I will provide today an introduction to not only the company, Collaborative Drug Discovery, but also the platform of CDDVault. So I'll start with a view, a different view than the one Dave used, but still a very nice view of the timeline of drug discovery. This one happens to be from the Eli Lilly website. And know that the CDDVault platform with data in processes and searching, mining, reporting capabilities, really just focusing at this point on this top layer of the funnel, where, as Dave mentioned, there's just tons of data that needs to be organized during this process. So scientists are creating and analyzing thousands to millions of different types of entities, like small molecules, cell lines, antibodies, or really whatever, throughout their drug discovery process. There's also hundreds to thousands of different types of assays being run over all of these entities. And these assays or protocols need to be devised, annotated, and really ultimately reduced to some definition for the kinds of data that's being captured for later analysis. Once that's done, we will be acquiring millions and millions, even billions of rows of data from all over the place, like CROs and partners, academic screening centers, in-house, et cetera. Sometimes this data is literally generated across the globe. Once generated, this data needs to be accessible to a collection of very serious, very thoughtful, well-educated, yet often stressed out scientists who need to be able to access this data in many, many different ways. Finally, this data is pulled together and summarized in all sorts of documents and reports containing both structured and unstructured data. So for decades now, we've all been used to various tools that we've acquired or built to manage the people and the data being generated in this process, from paper notebooks to flat files, various desktop tools, complicated mainframe database applications, and so on. Really working with all of these tools has resulted in an array of very unique challenges. But, of course, we feel there's a better way, a new way of doing things now, and that's what CDDVault represents. CDDVault combines many of the features from all of these previous tools into one easy-to-use cloud-based platform. With zero footprint, it is an intuitive system to use and requires no IT support. Training is provided, and customers are taken to full deployment, often within a matter of just 1 to 2 hours. CDDVault provides a variety of feature sets showing here for the registration of your entities and biological assay data, and an inventory system to help you keep track of compounds, a visualization tool where you can analyze the data, and an ELN for documenting all of your research, including the unstructured data that you are collecting. As we go through the process with our customers, we will get them up and running very quickly. There's a minimal training required, and you have control of your projects and protocols. The system is very easy to use. Since it's intuitive, there is a very high rate of adoption among the scientists and users. And with security built in, your data is protected, safe, and secure. CDDVault also grows and evolves with you. The support team can do more than just figure out which buttons to push. We also help you understand the best practices for handling your data. The system scales and grows and even provides an API, and that's an application programmable interface that allows customers to connect with the rest of their ecosystem, such as we've done here with Certara's D360 system. Just a quick note, collaborative drug discovery has been in business since 2004. We've always been based in the cloud, and through our partnerships with organizations such as Certara, we do provide a full solution for discovery informatics. So that said, I do believe at this point I will jump right into the system. I am logged in now into the same CDDVault system that is used by our thousands and thousands of scientists throughout the world every day. I start with a report such as this because I do feel it shows a variety of the types of data that our customers use and capture within the CDDVault platform. For this presentation, my first column here is small molecules. However, we do support the capturing of other types of entities like I mentioned earlier. So even if you are not working with small molecules, CDDVault platform will provide the mechanism for registration of other types of entities and then the association of all of the data you're generating as you move through your process. In addition to that, you may register additional data points such as synonyms and other data for your entities that you're registering. In a kind of global collaborative environment such as what we work in, it's very important to know that other organizations may call the same entity by different identifiers, and you can easily track those throughout the CDDVault platform. If you are registering chemical structures, we will also give you a set of chemical properties based on the structure you register. Just showing the molecular weight, but we have many chemical properties that are automatically generated based on the chemical structures that are registered. Kind of the middle here of this report shows a series of batch fields. Again, these batch fields are available for your users to populate during the registration process, and they are under the complete control of our customers. They will decide which data fields are available to populate, and they will enforce the business rules across those data fields. So a Vault administrator from customer sites can decide which data points to allow, which data points to make required, and even which data points have to contain unique values for each batch registered into the system. Now, these data fields can be text and numbers, dates, they can be drop-down pick lists or controlled vocabularies, and even file attachments, as I'm showing here in this column. For file attachments, if they are common image files or PDFs, we will show a nice thumbnail. Otherwise, we will just show the file name. It'll be blue and underlined, and users will understand that they can click on that file and open it on their computer. Another point of interest is this integration of an ELN, the electronic laboratory notebook, directly here within the platform. So you see I'm looking at a example small molecule that I've tested, but I can also see here there's a link out to my ELN entry. And if I go to that ELN entry, you can see that it is a synthesis ELN entry where I have synthesized that compound, and I have even directly registered that product into my database. So you can see here when there's an ID, that means I have registered that from my ELN entry into my registration system. And that's providing that two-way cross-reference link. So within our ELN system, of course, you can have text in different sections here. We do provide a full integration with Microsoft Office as well. So you can drag and drop Office files or other data files. You can hover over those. You can open those for collaboratively editing the documents. You can expand those to show the actual contents and so forth. So here's an example where I have some viability data that I have generated, and I have decided to show that in an expanded view directly here within my ELN entry. Clicking on this icon here would allow me to open this Excel document in Office and directly edit those right here within the ELN. So it's a very nice collaborative tool for sharing information about the drug discovery work that you're performing here. So I will go back now to my report and continue on. If I now look more towards the right side of my screen, this is where we are tracking the assay test results data. So in CDDVault, our customers are creating their own protocol definitions. I'm showing two different IC50 studies where you have IC50 data, dose response curves, and so forth. All of this data is organized into a series of runs. So you might see multiple run dates, which correspond to the actual experiments in the labs. Again, all of these protocol containers or protocol definitions are absolutely under the control of the customer. They do not have to wait on CDD or our support team to roll out any new data tables or anything to be able to deploy a new type of assay and immediately begin capturing the data that's being generated for that assay. They're able to create these protocol containers and start populating them with data right away. So this is the nice report here. How did I generate this report? So I will scroll up and talk a moment about the searching features within the CDDVault platform. It is a multi-parameter querying tool where customers can generate searches across chemical structures, either substructure or Tanimoto similarity queries, searches across the chemical properties that we calculate, searches across the assays or test information that is being populated for those entities, and other types of keyword searches, any of those data fields for the batches that we've talked about that the users are populating during the registration. So users can very easily and quickly create multi-parameter queries to search and mine and report on the data available within their CDDVault platform. In this example, I have done a substructure query for a particular fragment. I've limited the compounds to those that have a molecular weight range of 100 to 600, and I've indicated that I'm only interested in those compounds that have been tested in both of my IC50 studies. I've narrowed the search down to more current experiments or run dates, and I'm only interested in those compounds that had an IC50 of less than 100 nanomolar. So all of those search criteria went together and found the data report that we started off showing. Another note here, I did want to point out that we do allow the customers to organize their data across one or more projects. And these projects are very important and form the kind of backbone of our collaborative nature here within the CDDVault system. If I go to the settings page where we create those projects and I expand the Kimball project here, you'll see that these are the users that are allowed to see the data that's stored in that Kimball project. So for example, when Aaron logs in here, they can see the data in Kimball. If I go down to my secondary project here, you'll notice that Aaron is not a member here. So when Aaron logs into my vault here, Aaron only sees the data that I have chosen or allowed them to see in the Kimball project. They don't even know that the project 2 is available within this vault. So you can organize your data across various projects that, again, you create in your private and secure vault, and then you decide which users are allowed to see that data. All right, back to our search results here. Now that we've talked about the query and we've talked a little bit about the report, there's several things that you can do after executing query. One thing is you can customize your report. If I click this, it will show me now all of the data available in my vault that I can turn on and off in my table. So if I was interested in the log D calculation, for example, and these are all of the chemical properties that we will calculate for you, I could turn on the log D. If there were other batch fields and data points that I wanted to allow, as well as other data, assay data, I could turn those on. And once I update my report now, all of the new data that I have chosen will be displayed in my report. Users can set up their default reports so that if there are data points that they always want to see, they are quite able to do that very easily so that whenever they do a search, their default data fields are always available. Once you have performed a query and customized your report, you can also save that search. Now, when you save a search, you're not only saving the query up above, but you're also saving the layout of this report. So when you execute that search next week or next month or in your project team on Friday afternoons and meetings, you may get more than 70 results because new data has come into your CDDVault that match your search criteria. But that data will still be presented in this format. The saved searches can be private for just the user or also shared in a project so that other users can take advantage of the queries you are performing. If you are looking at raw data, you can also flag outliers directly here within the table. We do have the capability of building a Bayesian activity model, and you can also create collections here within your CDDVault. These are collections of molecules, and again, those collections of molecules can be created privately so that only you have access to them. But more importantly, you can share those within projects. And then once you've created a collection, you can actually share those with specific users really to enhance your collaborative experience. And then you can also export data out into XLSX, CSV, or SDF formats. So this is a good point here where we have gone through the search, we have gone through the data report, so we have talked about access to the data in part of this drug discovery process. If I go and click over here on a specific molecule, I just chose the first one from my report. I call this kind of the molecule report card here. This is all of the information or data that I know about this particular compound that's stored within this vault. So again, you can see the synonyms. If there were any molecule level data fields, they would be available here, and also all of the chemical properties that we calculate because this is chemical structure was provided and registered. You may have multiple batches here, so if you register this chemical structure multiple times, we will do duplicate checking, and if the same structure is registered twice, we will absolutely give you multiple batches for that same compound. Here again is a link back to that ELN page, the synthesis ELN document where this molecule was initially registered from. And again, if you were discovering this compound through a search like we have done and had clicked through on this compound because it was somehow very interesting to you, you would see this link and know that there is even more great information within this ELN about this particular compound. Again, all the data fields that the Vault administrator has made available for the users, numbers, text, dates, tick lists, control vocabularies, file attachments, all of those are supported. As we scroll through the various subtabs here, you will again see all of the other data that is available for this particular molecule. The Links subtab will show any ELN entries or any links that have been created for this compound. If there were any plates that were screened, you would see those in the Plates tab. Any assays or protocols that have data for this compound is here. You can click through and see the actual data points that are available here for those assays. If you created collections and one of those collections or any of those collections included this compound, you would see those listed here. And it's usually pretty important to know which projects have access to the batches of this compound. So this Projects tab will always show you really which projects this batch or these batches of this compound have been shared across, and you would know through those project associations who has access to this data. And finally, you may attach files to the parent structure itself. So in this case, I've put in an MSDS for this particular CC318 compound. So that is a quick tour of the CDDVault platform, and I think I will now stop sharing my screen and hand this back over to Dave to continue our presentation today. Thank you. All yours, Dave.
Thanks a lot, Charlie. So great overview of what CDD offers. And I think the real key part there is the flexibility, simplicity of getting all that data in. Just in preparation for today's webinar, Charlie gave me, I think, a 15-minute tutorial, and I managed all on my own to get all of that data in so that we could then view it in very different ways in D360 for thinking about analysis. So just before we take a look at D360 itself, I just wanted to say a couple of words about Certara and give you a general introduction to what D360 is and does. So Certara itself, the goal is to very much enhance the whole drug discovery and development process and really to improve patient outcomes. And so Certara itself has a wide range of technologies and services that support different aspects from PK and regulatory writing to detailed modeling of data and real-world evidence. Today, however, we're very much going to focus on the informatics software portion. So just to look at what D360 is and does, the goal of D360 is always to improve the utilization of data, really to take information and get a faster time to insight. When we look at how D360 is used and deployed today, there are a very wide variety of scientific research workflows for chemists, for biologists, for peptide scientists, for antibody research that D360 has been applied to. And I'm just going to show you one of those paths today. But the key to faster time to insight is really threefold. One, getting access to the data in a very timely manner as it is available. And we'll see in a second that when we look at D360, we get all the data that is currently in CDD. The second piece is just getting to the data is not enough. The data needs to be in the right kind of view, and we need to have the right kind of analysis tools to understand what it is about the data. So in our workflow for DMTA, we're not necessarily interested in the most active compound. We're interested in the compound with the best profile. And we're going to learn to look at all the data we have to see how we can take that information and design new compounds with improved profiles. And finally, research is not done alone by single scientists. It's done in teams. And so we need ways to share data and information, whether it just be the view of the data, whether it be comments and prioritizations that scientists want to add into this. And this applies whether we're looking to prioritize compounds within a series or design new compounds or look across different chemical series. But to really utilize all that information that has been valuably collected and entered into CDDVault. What are our goals for the product? So D360 is a very user-driven application. We allow users to file enhancement reports or requests within the application. We work very closely with all of our customers to understand what they need as scientists in terms of the data that they utilize. So our first and foremost goal is to serve that scientific need. And we also have an annual user group meeting and a customer steering committee to make sure that this is a formal part of our development process. We also deploy D360 in a variety of different organizational environments and also on different kinds of data. So it might be small molecule discovery, it might be antibody discovery, it might be preclinical safety. And everybody's database may be different. And so we need an extensible, configurable informatics system. But that's very much enabled us to very easily just configure ourselves against CDD and provide a ready-made solution that can be deployed very quickly. And as we've alluded to earlier, we want to ensure that there's not a high cost of infrastructure and IT resources that are used for this subject. So that means, look, the data is automatically available to the user as it gets entered into CDD. That's very straightforward when it's registering a new compound, but we also extend that to the availability of whole new assays. So if somebody puts in a new assay into CDD, that new assay is automatically available to users in the D360 side. If we look at where D360 is used today, I've already alluded to the wide range of scientific workflows, but it's also deployed from very largest customers, top 10 pharma that use it enterprise-wide, down to very small companies that might only have 5 or 10 individual researchers. And we have over 6,000 users worldwide. We can also apply D360 to a wide range of different kind of modalities. Although what I'll show you today is chemical structure centric, we have applied D360 in a variety of ways and have tools for analyzing the structures of modalities beyond the small molecule, all the way, in fact, into preclinical safety. And we also support cross-organizational collaborative research. So where you might need to make some data, but not all data, available to a partner, that's supported by D360 and its add-on capabilities. Just in terms of practical usage of D360, most usage begins with one click. We have something called a widget, which is a simple facade on a data query. That might give you a standard project view. Click search. That goes off, it gets all your data, but it can also contain additional automation steps to format that data, to present it in the right precision, to color it, and also to create those visualizers which you find most valuable. So that's what most people do with D360. The key capabilities that the application actually has is it can connect across multiple data sources. So the CDD connectivity, that just is one of the data sources, but if you want, you can connect it to a variety of others in addition to that. Self-service data access is vitally important. All scientists can build their own data views for spreadsheets or forms or other visualizers. It's a simple drag and drop interface. It provides all the same capabilities as Charlie described for CDD, but also some additional ones such as how is data aggregated and potentially pivoted when assays have conditions. but all very straightforward. To build the queries that you'll see in a second took about 2 to 3 minutes for someone who's reasonably experienced. We also have a lot of data formatting and visualization tools, which I would say are general data analysis capabilities. But then we have more specific analysis tools for small molecules to allow things like R group analysis, to do clustering, to do chemical series definition, and to do predictions of contributions of R groups to biological activity. And all of the data views, queries, etc., are shareable through a workspace system, very similar to the project that Charlie described earlier for CDD. And then we also have a secure partner application that provides the same level of analytics capabilities, but can be deployed to external research partners where they have limited data views limited to those things which are secure. And of course, we also inherit all of the security settings within CDD to ensure that the right people see the right things. So let's actually just have a look at the G360 side of what Charlie showed for CDD. So here you can now see my G360 dashboard. So each of these, so the key part of this dashboard are these widgets. So here in the center of my column is one called Kinase Project Overview. So this is a data view that is essentially being saved into a Kinase project workspace and therefore shared to every member of that workspace. So for me to go and get my project data overview for the Kinase project, all I need to do is click search. And that's going to run a data query against all the data from CDD. I've picked a subset that is specific for my task. And then it's going to generate, in this example, a spreadsheet-based output with formatted data, and also some other visualizations that I find useful. So here in the spreadsheet, each individual row represents a chemical compound, because my goal here is going to be to design new chemical compounds, and I want to understand what the data for existing compounds is like. If we look at our biological data column here, we see here's an IC50 value in nanomolar for the CSRC. And here we're also aggregating on the fly. So when a compound's been tested multiple times, I really want to understand, okay, this compound is, what's its average activity when I look over those? And geometric mean is a good way to do this. We also have some little in-cell indicators here to give an idea of the quantity and quality of data. So here, this compound has a mean IC50 of 37 nanomolar, but I can also see in the bottom corner that the 37 comes from 3 different values. And anytime I see aggregated data, I can double click on it and look at drill down. So here I can see that, all right, this compound's been tested 3 times. It gave a value of 37 nanomolar, and 44. Oh, and here it was batch 2 that was tested. And I have all the other meta information about the tests and the runs and the dates and the people that were available from CDD. So all of the data here is aggregated on the fly. We also see a number in the top right here, which just gives me an idea of the coefficient of variation. This is a nice small number, in this case, 18, meaning we've got nice tight grouping of our 3 values, and this 37 nanomolar is a believable number. Now, this data set is also sorted by how recent the data was created. So this is my most recently tested compound. And we can also see that these numbers here in the purple and underlined italics, those are saying these are the new data points. So this is what Charlie just loaded up in CDD, and then I've set it to be highlighted, new data to be highlighted in G360. So here now, we're starting to get an overview of the data we've got. So here is our primary activity data. These are selectivity assays. So we want low IC50 values, high activity here. And over on these, we want high IC50 values, and these are colored accordingly. So although this compound has an activity of 10 nanomolar in our primary activity, it's not very selective for either our HCK. Sorry, our HCK is the wrong way around. Just swap that. It's not selective like we'd want. We also have some other default data views here. So I've got a quick histogram of our primary activity. All of these views are interlinked. So this shows us where our primary activity is high. Here we have a plot of a multi-parameter score. So one of the columns in my spreadsheet is a multi-parameter score. So in order to explain what this is, let me just sort by our primary activity and get the most active compound at the top. So this is our most active compound at about 0.12 nanomolar. Again, it's been measured 3 times, but a very large number on the top says, hey, there's a big spread of numbers. If we drill down, we can take a look at that, and sure enough, 158 nanomolar, 0.12 nanomolar, there are many orders of magnitude spread. So although this looks like a highly active compound, we cannot be sure of this number. But also we're not looking for activity here, we're looking for profile, and we can see here that we've got activity of this compound in many of the other screens which we don't want. If we look at our multi-parameter score, this actually balances our needs across all of these assays to have a low number here and high number here. So if we sort this in descending order, so we get our best scoring compound to the top. So now here we've got a score of 92.3. We've only got 37 nanomolar in our primary assays, but we are nicely selective. So this is our best compound. And actually, that's what this plot down here shows. So the center of this plot is our ideal profile, and points are plotted close to it depending on how similar to that profile they are. So this is our best compound. If we look at compounds that are further away, these are about 60% similar to our profile, our ideal, but they all have a similar profile to each other. And in fact, we can see here that they tend to have poor activity in the primary assay. If we look over the other side, these compounds, again, they're still 60% similar to our ideal, but they have a different problem. They have a selectivity issue. So these plots tend to guide us in order to be able to develop new compounds. But let's actually do some compound design. So what I'm going to do is, first of all, take a divide and conquer technique. Let's do an R group analysis so we can start understanding our R groups and how they contribute. So I'm just going to sketch a quick core here. We're integrated with ChemDraw to do this work. So we will choose our R1 for our R2 and remove the excess. So this is now going to be our core. Here's a core structure. Let's just analyze our structures to make sure we've got compounds that have those cores. Yep, 198 compounds have it. That's great. So we'll add our R groups to our data set. Now, if we scroll over, we see our R group columns. But let's have a look at the R groups and how they combine versus our activity. We'll do that just by creating an R group matrix. And I will just move this to a new tab to get a better view of it. So here we're seeing all our R2s on the x-axis versus our R1s. But which of these compounds have good profiles? Well, let's color it by our MPO. So just search MPO and we'll apply a color scheme. So now our green points are the good ones, but this is still hard to tell where are our good R groups. We can make that much easier by just reordering our axes. So let's go to our axis, say, all right, x-axis, we'll sort by MPO score. Oops. Cancel over that. Our x-axis will go and sort by MPO score. And we'll do that in descending order. So apply. And we'll do exactly the same thing with our y-axis. Now, what have we done there? Now, all our R groups that tend to give good profiles are now near the origin. So we can see immediately that we actually haven't created many of the compounds that would seem to be the best combination. So what I'm going to do is just select a few of these R groups. And what we're going to do is enumerate these compounds, essentially the ones that should be down here. So we'll just say enumerate. Here's our core that we drew. Here's our 2 R1s that we picked, our 2 R2s. And what we'll do is just create those new structures. And so 2 by 2, we're going to create 4 compounds. Let's add those to our data set. So now if we jump back to our spreadsheet, right at the bottom, we have 4 new, what we refer to as virtual compounds. Okay? So we've just used G360's analytics capabilities to look for compounds with good profiles and then enumerate those things which have good R groups. So what I'm going to do now is send this over to Charlie so he can create and register these compounds. So I'm just going to save this data set. So we'll just say save as. We'll put it in the kinase project so Charlie can reach it. We'll save that. Now we're done with our design process here. So I can just jump back to my G360 dashboard. We can take a look at that workspace. And here is my set that I want to send to Charlie. So I'll just email a link to this data set. Send it to Charlie, and he'll now get that information. Now let's see how this process completes, and I'll hand back over to Charlie. So Charlie, all yours to do your little registration.
Thank you, Dave. Thank you. Let me share my screen here. All right. So I received that email from Dave, logged into D360 and grabbed that nice data set. What I've done is made myself an SD file containing those 4 virtual compounds that he wants me to load into the CDDVault and then send for synthesis. So now back in the CDDVault, I'm in the Import Data tab, where we go through a quick 3-step process where we choose the data file that we wish to import. We map the fields from that file into our vault appropriately, and then we get a validation step where we either choose to commit or reject the data based on some QC report that we receive. So here I'm going to choose that SD file. We could also use Excel file formats as well, but those are the virtual compounds. And I can put this straight into the Kimball project or put it into some other project for maybe virtual compounds, so I have those kind of separated out from the rest of the compounds already in my CDDVault. So I would choose that project and load that SD file up. Now we get some options on what type of data are we importing. So are we importing assay data, updating existing compounds, or in our case, we are registering new molecules, and that SD file does have chemical structures. Down here, we get a representation of what our import file looks like. So it has a mole file, which is the chemical structure, and he also provided that VC number coming out of D360. So I can map those columns into my vault. So this section are all of the data fields in the CDDVault where I can map incoming data to. We have a guessing algorithm, so our algorithm has decided or guessed that the mole file is probably the chemical structure. That is a perfect guess. I will click apply. It also now thinks that the compound name is a likely synonym. Now, if it's not, if I had some other field created, I would just navigate and tell this import exactly where to put this data. But I do want to include that information here as a synonym for my new compound, so I will apply that. This guessing algorithm gets better and better. There's a learning component to it so that the more files you import, the better our guesses are. And then you can also save your mapping that you've performed as a template. So if you know you're going to be getting compounds like in files like this again and again, and you want to map them in the same way, you can save a template so that the next time you import a file like this, you can do all of this mapping and import in truly one click. So it makes it even more efficient than it already is. So I'll process this data file. At this point, it's making sure numbers are numbers, text are text. It's making sure those business rules that your Vault administrator has set are being met. So required fields are there and so forth. It's also making sure that we can interpret those chemical structures appropriately. And we come back with this QC validation report that says if you hit commit, you will get 4 brand new molecules registered. If we had other sections, they would be broken out. So if you did have some errors or other suspicious events, you could do that. And you can even choose to accept and reject pieces and parts of your import. So if you had some compounds that were new molecules that you wanted to accept, but other compounds that were already registered that would report here as new batches, you could reject the new batches because you were thinking, gosh, I didn't realize I had already registered those. So at this point, I would commit that data import because this is quite lovely and just perfect, as you might expect for a webinar situation. We planned that very well, Dave. Well done. So we're processing this, and in a moment, we will get a commit report that says, yes, your data was committed, and I can immediately view that data by clicking one quick link, and you will see all of the 4 compounds here that were registered here. Now, I can add this to a collection. So new compounds November 5th. I can put this in a project, and once I save those 4 new compounds into my project, I could now immediately share this collection with any other member of that project. So I could now, well, let's not choose Dave because he gave them to me. So I'll go ahead and send them to Ian, and I would say, hi, Ian, please synthesize these new compounds ASAP because they are indeed amazing. So what Ian's going to get, you would type better than I did. What Ian is in the end going to get is an email with this link. No data is being emailed outside of CDDVault. Everything is safe and secure within the platform. But Ian gets this link. As long as Ian can log into this vault, he will immediately come to these 4 compounds and when they take them through his synthesis workflow. Now, I'm not going to post this message because Ian will be sending me an email pretty quickly going, what are you doing, man? So that's pretty much it. In summary, I'd just like to say CDDVault allows the users to store their data, import their data in this manner, or even integrate or automate that data import through our API so that the data is then available. And through our integration with applications such as D360, that data becomes available for additional analysis using any of the tools in your ecosystem. All right, thank you very much. I will stop sharing. And at this point, I believe we are going back to Dave for a wrap up. Perfect. Thank you, Dave.
So just to conclude here, so what we've been talking about is how CDD can help capture all of data, do it very easily. Anybody can use it. It's secure, but it's also collaborative, and that really helps with our data in portion. G360 really helps with our analytics and data out portion, the self-service data access, the automation of a lot of the capabilities in that. And we tried to emphasize this particularly today with looking at this design, make, test, analyze cycle. Now, clearly, we've used particular data today. We've looked at IC50s, but we could have looked at any kind of data at all, whether it's percent inhibitions or ppm measurements or PK data. All of that is supported through the system. We just wanted to keep it simple and focus on one particular area today. Also, we've looked particularly at things probably more interesting to chemists. There are a whole load of biologics workflows as well that are supported through the system. But what I really wanted to emphasize now is this best practice now of thinking about your data in and your data out together in order that you don't go through these periods of, well, I've captured my data well, but it's hard to get to it, or it's hard to analyze it. Thinking about these things together is really what shortens those drug discovery cycles and gets you to where you need to be faster. And working together, we've created this thing that's all cloud hosted. It's all pre-configured. There's essentially just the key work to do is to turn it on and make sure people understand how to use the software. So the key part for us is this is a very exciting partnership because now we can work much better together to take advantages of the specializations and skills of both organizations to help people in their actual research work. So that really concludes what we're going to talk about in terms of the webinar, and I think we might maybe just have a minute for Q&A.
Actually, we're right at 12 p.m. Eastern, so we don't have time for a live Q&A. We did receive several questions during the presentation that we will address via private email to those of you who did submit a question. So be on the lookout for an email from our sponsors to address your questions individually. I'd like to thank our speakers for today, David Lois and Charlie Weatherall. I'd also like to thank our sponsors, Certara and Collaborative Drug Discovery. Finally, I'd like to thank all of you for attending today. We appreciate you choosing to spend this time with us, and we hope you gained some valuable insights. Stay safe, and we hope to see you at another BioIT World web seminar soon. Thank you. Bye.